So you have a desired donor gene, and you select it, you take it out, and then you isolate it, and then you try to put it into a vector. And a vector would be a virus or a plasmid usually. And we've worked with the plasmid before. And then the host is usually a bacterium or a yeast that can be replicating the gene and translate it into a protein product. That's why I said heard about that before. So what did we do? What did we do? We didn't actually cut it, but we had a vector, which was our vector. What did we use to have a protein expressed by bacteria? You did it right over there. It was expressing while you were watching. So that's the protein. What did we use? Did we use a virus to bring that into it, or did we use a plasmid?
To put it into, or
to
put it in?
Well, we had this phase of.
We used the bacteria phase to put it in a bacteria, right?
No, we used the remember the vector, the round donut-shaped thing. Yeah, so we had the protein in there, and then we had something else in there we'll talk about. I won't give that away. So it was a plasmid. And when we talked about that, how that's DNA that's in the bacteria. So bacteria have their main double-stranded round chromosome, but don't confuse that with the plasmid. So the plasmids are just extra. A lot of times they have resistant genes on that. Yeah, remember the round one, and we have those different sections.
What does the bacteria normally do with the plasmid? For extra stuff?
Yeah, there's resistances are on there. There's just I think they probably in ideal situations where they wouldn't need some of that, they would probably be okay without the plasmids.
They always have them?
Not always, but a lot of them do. And because they're smaller and we know a lot about them. Remember we talked about like how many base pairs, the shortest one, really short ones. They're ideal candidates to put something in that you want to bring into another cell. And did we use E. coli to transfer it in? So we basically had already that cut out. We already had a plasmid. So the plasmid was the vector. The cloning host was E. coli, and then it produced the protein for us, which was the fluorescent. So it's what we did is again shown here. You have some cell, whatever cell you're working with, you're interested in a certain gene, you take that out, you cut that out with your restriction enzymes, and you insert it into a vector, in this case a plasmid, and then you can actually have this bacteria grow, and so it would produce a lot of that product, and then you can do certain things. You can have those bacteria, for example, instead of fluorescent, it'll produce your insulin, it'll produce your human growth hormone, it can produce your vaccines. It can also, if you put it in a plant cell, it could actually produce transgenic plants, which are the genetically modified GMOs organisms. So it could include that gene you're looking at, could, for example, have a pest resistance. It could include a herbicide. It could include it can actually increase the if you know that there's a certain protein that's important for a specific, or that's important to our health, you could include that. And I heard of, and I just remember that now, and I don't know if that's actually an example of this, but I had this friend, she's Mexican, and she went to Bangladesh. What do they eat in Bangladesh? Rice probably only, or they have one main food group that they eat, but they had a deficiency of some vitamin, and so basically what they did is they said, well, if we can't convince them to eat something else, because sometimes it's just like with the patient that doesn't want to take the antibiotics every day because Thursday is Thursday, and you have to stop. Exactly. That's going to be a really thirsty Thursday this Thursday, right?
Real thirsty.
So instead of doing that, they basically decided to add a protein to, say, the rice plant to have them eat exactly the same diet, and now they would have that vitamin in their diet that they use, that they have a big deficiency. So that's another option. So it has a lot of benefits. We can also use this, and we already talked about to look at gene function, sequencing, identification. So that's more the technical, the more the genetics part, and this is more for what they're now used for. So it's pretty interesting stuff if you think about us being able to make to utilize bacteria to produce whatever we need, little factories. An important two things is, so the vector, and we use the plasmid, is must be capable of carrying a significant piece of donor DNA. So the protein a lot of times is longer. If you have a plasmid that's very small, it might not be able to incorporate that gene. So the vector, plasmid, or the virus needs to be able to put this gene in, and then it must be accepted by the host. So if you have a plasmid and it doesn't enter the E. coli, you're never going to get E. coli to produce the fluorescence that you want. So plasmids, we worked with this one on bacteriophages. They're just interesting to us because naturally they inject the DNA. Important difference between an animal virus and a bacteriophages, that these ones are always these are the viruses that infect bacteria. That's the first thing you always have to write down to ask for that. And then they inject it, whereas usually the other the animal viruses fuse or endocytosis. Remember those different ways of getting in? Yes.
How do we get what we want in the bacteriophage?
I don't know specifically. I haven't had a lab on that. I would have to guess right now. It's probably a somewhat same simple technique where you say maybe the viruses, you have an infected, you have viruses in a cell culture, for example, and you throughout assembly of the virus, I don't know if there's anything coming, but they basically integrate a gene into their genome just like a bacteria cell would, you know. So when they assemble in a cell and you give them a lot of genes or nucleotides that you want, and you give them a high concentration, they might integrate that into theirs, and then you can select for them the ones that did it, and when they multiply in the next cell they infect, you get your DNA constantly remade. So this is a typical vector. As we said, it must accept the DNA, the right size. Origin of replication is needed. We had that too. Remember, we had a place where the process of protein or the process of reading the protein or the sequence for the protein was done. And then important is a lot of times they include drug resistance into this. Do you remember why? Why was drug resistance also in this vector? Say the vector is, so this plasmid has all kinds of stuff, but there's here now 2 resistances. What does that help us with in the long run?
So you can select aptolins and then you get the vector.
Yeah. So then if you put them on the plates, man, I thought that too. Yeah, I guess that's what this one is. So then if you would put them on the plates and you have a selective medium, which means it has the, say, ampicillin in the medium, then it can't grow there, and only the ones that have the plasmid with the resistance can grow. So you select out the ones that are successfully transformed, and these ones aren't. We'll get back to that. So that's the main reason why you would include that, if you can, into your vector.
So it helps you with the selective process?
Yeah, it makes it easier. You could probably, I mean, it's teasing out where it happened and where it didn't. So in our case, you would probably say, well, we had not only selective medium by adding ampicillin. Did we have that in 2 of the 4 plates we had, or did we have that in more? Because there are a couple of different steps, but it helps you to determine which ones are successfully transformed, which ones did take the plasmid in, and which don't. And this is really just, easy for you to understand. I think it, what you would want from a host like E. coli, you want them to grow fast, so they produce a lot. You want them to grow in large quantity. Ideally, in a normal culture, you don't want them to be pathogenic, because if you want them to be easy to work with. You don't want to walk in with I don't know how many safety and security issues. It's good if the genome is well understood, so that smaller organisms, smaller genomes is easier. They should be accepting the plasmid, obviously. So E. coli was perfect because they were all. I mean, I didn't even see. I maybe saw 3 cultures that weren't, or 3 cultures that weren't transformed, but most of them were. It should keep producing over multiple generations. So when they replicate, how do bacteria replicate? It's the only way they do. Not the only one, maybe, but the main one. Do you remember the 2 sister cells, binary fission? Did you say that?
I was thinking it.
And then they should produce a lot of the whatever you put in. That's important too. But these are all clear. If you don't have to study that kind of thing. You can just think, well, what do I want from a host cell? What do I want? I want product. Okay, I want them to produce a lot. They have to replicate fast. I don't want them to be I don't want them to be pathogenic, because then I'm going to have a problem working with them. Things like that. So it should be a line of cheap at the bottom. The usual characteristics of desirable, cheap. So what we do usually, what you can do is, and we've seen that before too. So you cut out. You have a gene that you're interested in. You cut it out with a restriction enzyme, and then you cut the vector, your plasmid, with the same restriction enzyme, because you want them to be able to attach to each other. So there's the restriction enzyme, there's the ligase, and then you have this plasmid. So now you have a recombination, a recombinant ring of DNA, and then once you get into the host, there's transformation. We've talked about, we've done that ourselves. Then there's multiplication, because you want that. There's protein synthesis. So once it's transformed, it can multiply, or it multiplies no matter what the bacteria does, or your whatever microorganism you work with. Then it hopefully produces your protein, like it did in our case, and then you purify that protein. That's the last step that we didn't have to do, but you could purify that protein. So if for example, you're producing insulin, in these beakers you have these bacteria that are transformed with the, if you go back, so say you put the insulin gene in there, it goes into a vector, the vector gets into bacteria, the bacteria is starting to read it, it multiplies and it starts reading it, so it produces all this protein, and then you purify the insulin out of it. And that's probably some process that you find that I don't know exactly what that process looks like, but there's ways to purify the protein that you want. So that's really why we use recombinants, just to have a gene expressed that's desirable, that's desirable because it codes for a protein that we want. And we talked about that. If you have that in here and you have a selective medium versus a non-selective medium, you can actually select for the ones that have the plasmid, because the plasmid not only codes for the protein that you want, but also for resistance, whereas in this case, they all grow. You don't know which ones are growing, or which one of the growing ones have the plasmid and which don't. Here you have with plasmid, without. On this side you only have with. So it's straightforward. Probably smart people behind this initially, but now it's like, yeah, makes sense. Don't know why we would do it any other way. Which of the following is a primary participant in cloning an isolated gene? We need a restriction enzyme. Do we need a host? We need a host. We need a vector. All of them. So these are these are the main components we need to.
What's ligase?
Ligase?
Oh, right.
Yeah, the sticky ends. So these are a few examples. Insulin for diabetes, human growth hormone for dwarfism, erythropoietin for anemia, or I added doping. It's always or improved, what do you call that? Yeah, let's call it improved performance. Slightly improved performance.
Performance enhancement.
Huh?
Performance enhancement.
You guys are all doing it, huh? The way you talk about it, it's just enhancing a little bit. I'm just helping my body out.
It's called caffeine.
Which is true in the end. That's what it is. Caffeine. No, you're not drinking anyway.
Not today.
No.
I used to have like a caffeine soda.
Other ones is factor VIII, so there's a factor for hemophilia. I don't know if the bleeding disorder, really rare, where you can, for any little wound, you would have no, there's a factor VIII is missing to clot the blood, so you would be able to give that, to have that produced, and people can inject it, vaccines, so we've covered most of those, and then I said, there's also the organisms like the ice crystals in the strawberries. So you really have that, and there's lots of interesting stories. There's some, for example, that encode for a insecticide. So the plant has an insecticide, and it colonizes at the plant root. So insects that are trying to get near the plant root are repelled or are dying. And you see that a lot now that, especially in plants, because you can't really control their reproduction as well as, say, other like animals. If you have transgenic, I don't know, mice, you can keep them and you know exactly what's going on, how you treated them. But a lot of times now, I think I've heard just not too long ago, some farmer in Oregon sprayed his whole field with pesticides because he wanted to like start something new the next season, and then like this one square plot just stayed straight up, and it's like, what's going on? Like, why is it not working? And it didn't work, and they analyzed it and found out he had some Monsanto, the main company that makes all the, they do that a lot now. They have plants that you buy, and you buy them with like a specific insecticide, and because their plants are resistant to the pesticide, then they grow, whereas everything else doesn't. And so he must have gotten one of those plants in there, and they spread, and they still don't know how it happened, because they had only tried those on experimental fields, and they were very far away, and it didn't show up anywhere else. And then the company, I think, said somebody stole it probably from their experimental area and planted it there, which is a really, really weird explanation. And then and then I don't know what he or somebody else, there were some other explanations how it could have happened. But we have even, people leave notes on your door. A lot of sunflowers, and they leave notes on the door, and they say you have some, wild type or whatever, and if you would allow us to take those out, because they're trying to breed a certain type in the area that is really like a transgenic type, for example, that's really successful, and then if you have some unsuccessful, I always have like 20 heads. Those sunflowers are not really beautiful, like they don't have one big head to produce a lot of seeds, but a lot of different heads. They don't want them to pollinate their plants and then get this wild type DNA in again. So there's all these problems now, because you can't really control the pollen. they'll find ways, I'm sure.
Like if your neighbor has a bunch of weeds in their grass,
Yeah,
and then they sprout and blow into your nice lawn.
yeah. Yeah, don't have a nice lawn, it's not gonna work out.
I know a guy that he would like to put his lawnmower
So
after you.
there's more and more of this, which is really important for production of different, especially especially for plants. And then we have also mammalian cell cultures that produce enzymes, hormones, antibodies, things like that. We use them for oil spills. I don't know if you heard about, introducing certain bacteria that can actually help in cleaning up oil spills. Or adenoviruses, for example, are known for causing the common cold. And then because they have, they can also recognize certain cancer cells or proteins. That virus is desirable as a vector to bring or to infect specific cancer cells and kill them that way. So that's and that kind of bridges over to what we're going to do at the end. Is these are ways of tweaking plants or bacteria to produce products that we want. So yeah, these, I think this is an example for the tomato where you have, it's called endless summer tomato. Used to be endless summer, now it's the endless summer tomato. So they're slower, they ripe slower, and they have increased acidity. So that's one of the things that they thought was there's different characteristics in plants that are desirable. So we're already working on, or the research is already pretty close to changing a lot of that, and that's where the GMO foods are, and that's where the discussion is what's good for us and what isn't. But it's definitely well established already. And then in animals, what you can do is, because you or in animal cells, you have different ways of getting you're injecting the DNA into a cell, and so what they usually use is what they call pronuclei. So these are early embryos, and so you have a nuclei in there, and you can actually put it on a plate on a culture slide, just like we use with a depression, and put it in there, and then you can see in this picture how a pipette holds the cell, and how this I don't know what the holding pipette, what they call the injection pipette, how that can inject the DNA that you want into this cell. So now you inject this DNA, and it can be incorporated, and then you take that cell and give it to like in mice, for example, and give it to a female, and then if the female produces an embryo, and then keeps the finishes the development of that embryo, you have a mouse with a specific trait that you wanted. And I think you might know what those guys were exposed to, but that's how far you can go. I don't know if that had any kind of what the idea was behind this, but besides knowing that it worked. So you can use a virus to transfect the fertilized egg or early embryo. It doesn't have to be this way. This is basically what what you can do in the laboratory. But the idea is that these animals are transgenic, and they transcribe and translate our genes. So, for example, if you look at them as disease models, you can experiment with them and say, you have models, and you can look at genes that you think are important in certain disease, and you can either have them included in their genome, so they're expressing a certain protein, or you actually try to take them to erase those and we get to that, how that works. But at any rate, you're trying to manipulate the animal genome to see how that's involved in certain diseases. And then you also have animals produce protein, for example, in their milk, and so you can do the same thing. You have goats or sheep, transgenic goats or sheep. They produce a specific protein. They give it off with their milk, and then you separate it out, just like out of cell culture. So even this is already possible. So that's what transgenic animals can do for you. Where's the number here? 37. Okay, 3-4 more slides, and then we'll have about after a short break. We have about 45 minutes still. So far, what we looked at is how do we introduce DNA into the body? How do we get DNA in so it produces a specific protein? But you can also, the problem then is, what if the protein that you have produced in somebody's, like in a patient's body, for example, is faulty or missing? So what else can you do? And you can actually, for example, when that's what gene therapy comes in, you can correct or repair a faulty gene in humans. And what you see here is you have you identify the gene that might be responsible for that faulty protein in a healthy human, and then you cut that out and you isolate it, and then you insert it into a virus. And I don't know how that exactly works, but a virus picks it up, and then you isolate from the patient marrow stem cell, so from the bone marrow, and then you have the virus infect that stem cell with that specific gene that person doesn't have, or has a faulty copy of it. And then when you reintroduce, I guess, the marrow stem back to the patient, then these cells would produce more cells, and they would all produce the right protein. That's the right protein from the gene sequence that you took out from a healthy patient, or from a healthy donor, I should say.
Like in what, like bone marrow? How would you get a normal gene from a healthy subject?
That could be from any cell, because most of your cells have, all your cells have the same genome, but then it depends on what it is. I guess mostly you would use where it's expressed. So, your liver expresses different proteins than your kidney or whatever, and all these different organs express differently. Every cell, that's another thing they look at, what are tissue-specific expressions. So if this is expressed so for the donor, it's not so important. It's just important to know that the gene is intact, because the donor probably has it or does have it in every copy of his genome, every cell. But then for the host, or for the person for the receiving part, you have to put it somewhere where a lot of cell production is happening from these stem cells, and so once they have the right copy in it, you get more cells produced with that exact same that has the intact copy in it, and then they can produce the protein for your body that is missing. Make sense? And there's just this difference of ex vivo. So you remove the tissue from the patient. In this case, it's the marrow stem cell. But you could also do it where you directly introduce it to the patient's tissue. So you would try that through injections, etc. So you're not actually removing cells. You're trying to do that directly in the body, and those methods aren't all, they're not foolproof. They're not 100%. It's always a question of, just like with our experiment, how well does the transformation, how well does that work? How well is the host receiving it? How much of it is not being received? How much is digested before it gets there? So it's not clear. It's not a that's part of the therapy is finding out how well it works in people, and it's probably different from individual to individual.
So I see all these advantages of genetic engineering. Why is it in such debate? Like, what are the downsides of it?
I think mainly because it seems like, I think people would probably say you're playing with the absolute the first level of everything. So if you have, if everything depends on your genetic material in your body, absolutely everything, because it makes sure each single cell of yours is producing the right thing. And so there's just tampering with that is something that people are really scared about, and they are, even if it comes to food, because I think it's probably time that's going to solve those problems. Maybe I don't know what if you think about, if we eat corn, we're ingesting whatever every single cell of our corn crop has, or whatever else plant we eat. So we're able to deal with it, and I don't know why that would be different in corn that is maybe changed to making bigger, or just is more productive. And so I don't see that either. I think it's really mostly the fear that it's a machinery, because it's at the base of everything. It's a machinery that we seem to not be able to. Well, we don't know all the consequences of it, I think is really what they're worried about. But at the same time, I think there's so much progress in the field that it's debated until, we don't know how to feed the world anymore, and it's debated until probably you have you see, especially for disease, you see really the need of finding a cure, and I think it's moving along pretty well. There's other things, for example, and I don't know that much about that, but if you need these embryonic stem cells, for example, there's always a question of, well, if you want to use, say, a human one, when is a human a human? Is it a one-cell organism? Has it to be a baby that's newborn? Where do we draw the line? Because you could say, well, as soon as fertilization happens, there's an organism. We can't do it anymore. And so if you want to use that, because that's the state where you can get all this, where tissue can, or where the cell can still become everything. It can become a liver, it can become a kidney. So you want to work with it, but then there's a whole debate of, what are you using for your research? Is this, where do we draw the line? So it's probably a religion in it, ethics, lots of other opinions that people have, or rules and guidelines they have, and they don't know if that works, or that if those guidelines are maintained and observed, or during those processes. This one I'm gonna skip because it's not really. I left it in, or I put it in because of the disease, but it works with RNA. It's a little bit more complicated. And then the last thing I was going to show you was when we've already done that, the DNA fingerprinting is one of the last parts of this chapter. It's just a method you use the restriction enzymes, electrophoresis. You hybridize possibly and do a southern blot, but you sequence the DNA of a person, because every person has a very unique sequence, especially in these repetitive areas. And we've gone through the whole process of, we've gone through a little bit less, but you would have 4 individuals here. They have the cells are colored different, so you see the difference, but you have 4 different individuals, so assuming they have different differences in their sequences, which they usually do. You cut them with the same restriction enzyme, so you have little scissors here. You put them on those 4 lanes, and you get these patterns. And this, I think, is in your book. It's an actual. You might not be able to see it here, but this is an actual. These are actually sequences they used in a trial. So you have evidence 1 and 2. Those are, I don't know where they found. It could be that they found 2 different. They found DNA in 2 different areas, and they call it evidence 1 and 2, but they're identical. So you know that whatever evidence they collected, it could be. I think it's a rape trials. It could be sperm. It could be blood. They find the same exact. There's these bands here. There's these bands here.
Suspect 1 is evidence 2, right?
Hmm?
Suspect 1 is evidence 2?
I think the evidence. I don't know what the.
Suspect 1 is guilty.
Who's guilty?
1.
Yeah. I don't know if you can see that, but so basically, if you look at suspect 2, because of this, you would immediately exclude that person and say, well, it looks so different from the evidence. How can suspect 2 be even a suspect? You got to send him home. And I think there's recent cases where people were really in jail forever, and because of those techniques and better resolution and everything, and just the progress they've made, they've been cleared after a long time.
Lots of people on death row too.
Yeah.
People that have already been executed too. Wow.
So you can use that fingerprinting, which we've just done. You can look at relationships in your family. You could look at like how related you are, hereditary relationships. You can look at disease, obviously, and see how that's inherited down the line. You can look at human evolution and try to find out, what, I mean, just by sequencing or by looking at these, how the genome changed over time. It's for identification. And then sometimes we've seen that before, where they, when you look at differences between organisms in evolution, you can go actually to the mitochondrial DNA and look at that also. Remember that the mitochondria has like a circular DNA, so you can look at that too, and that gives you some additional information.
Is that a plasma that they have, or?
No, it's related to the endosymbiotic theory that they have the regular chromosome. They're only, yeah, it's a circular chromosome, just like the bacteria have, and they are a lot. I think they're always from your mother, as far as I remember. So mitochondrial DNA, a lot of times you can look at that and compare different organisms and look at how related they are by looking at the mitochondrial DNA. Okay, and then if you want, you don't have anything, right?
I do.
Oh, you do?
So this is chapter 10.
Yeah.
And so what do you think that the question, like the important things are?
You said to be decided on the study.
So, I mean, what we talked about today, probably closer to what we've done and what we've ever exposed ourselves. You know, well, here I need a few more questions, maybe 6 or something. But do you want to talk about how the whole thing looks in 5 minutes, and then that's part of it. Here I would say, what did we talk about? Just the main techniques that are involved, and just what they're for. What is PCR for? What's sequencing for? What do we use these techniques for? Maybe knowing the what transgenic or what recombinant is. You want to slice it out? I mean, you want to. Good. Now I'll try to make it closer to what we've worked with, because that you have to review anyway for.
Is this gonna be on our lab final? Would this be more?
Yeah, I
just
mean,
the lab
because
instead of the written, maybe?
I think we can talk about the lab final tomorrow in the lab, but how we're gonna do that. But there's gonna be what we've done is it's a practicum, but shorter because there's not as much, I think. But then there's gonna then there's gonna be some multiple choice questions about everything, and there's more simple. There's multiple choice about gram stain includes A, B, C, or B, C, G, or you know what I mean, like the basic things. Yeah, but it's gonna be general. So I'll talk about tomorrow's, and if you want, you can look at your third chapter test too.